So in this video we will discuss about our second chapter, that is biological classification. Coming to our introduction part, so again the unit 1, biodiversity in the living world. In this we are discussing the second chapter. From this chapter we can expect maximum 3 questions. Okay. So as I already told, biodiversity is life, diversity is variety of living organisms, variety. So it is variety of living organisms present on earth. As I have already told you that nearly it is estimated that 15 to 30 million species are there on the earth, out of which only 1.7 million species have been identified, have been classified and have been named. So depending upon certain features of an organism's, classification has been done. But when first the classification began, when first the scientists start to classify, they did not consider any scientific basis. They just started to classify organisms based upon very primitive characters, very primitive things they considered, like how they are useful to man. Some are used as medicines, some are used as fruits, some are used as vegetables. So first classification was based on this certain very basic characters upon which how the plants or how the animals are useful for human needs. On that basis the organisms were classified, but there was no any scientific base for this. Then the next was Aristotle was the first to classify organisms based upon their scientific basis. He considered some scientific aspects to classify the organisms. For example, under the morphology he considered herbs, shrubs, trees based upon its habitat, based upon its habit. They considered or they classified the plants into herbs, shrubs and trees, and also has identified or he has classified animals into animals which are having blood and animals without having blood. So this was the first attempt which was made to classify organisms on their scientific basis or some of the scientific aspects. So as the days moved on, even this system has lost its importance again. Next system was upgraded. That system was Linnaeus, who proposed a 2-kingdom classification. Linnaeus, he proposed a 2-kingdom classification where he gave a mention of plant kingdom and animal kingdom. He grouped all the plants under plant kingdom and he grouped all the animals under animal kingdom. So, but one thing is, he did not differentiate between prokaryotes and eukaryotes. He did not make any classification on the organisms which are prokaryotes and the organisms which are eukaryotes. He did not give any mention on this. Then he did not consider the cellularity of the organism, whether they are unicellular or they are multicellular, and whether they are photosynthetic, whether they are not photosynthetic. All these aspects were not considered. Very small scientific base was considered, and based upon that the organisms were classified. So Linnaeus gave 2-kingdom classification. As the days moved on, even this has lost its importance, and again the next scientist is called R.H. Whittaker, who proposed an ultimate system of classification which is even in use till today, that is a 5-kingdom classification. He classified organisms into 5 kingdoms. Those classification was based on their organization, how the structure of cell is, what is their mode of nutrition, what are their phylogenetic relationship, and what are their reproductive characters or reproductive structures. He considered all these aspects. Especially we need to talk about here is about evolutionary relationships from, say for example, is this organism is here, now this organism is here from where this organism has arrived, who are their ancestors. So in the evolution part, who are their ancestors, from which organism this organism gave or took their birth, this aspect they have discussed, that is phylogenetic relationship. Next one is reproduction. So this is very important aspect, or this is very important point. There may be a chance of coming questions from this point also. On what basis R.H. Whittaker classified the organisms into 5 kingdoms? On their organization, cell structure, mode of nutrition, phylogenetic relation and reproduction. And considering all these aspects, he classified living organisms into 5 kingdoms. Those kingdoms are kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae and kingdom Animalia. See here, kingdom Monera, then nothing but bacteria, next one is Protista, next is fungi, Plantae and Animalia. First we will go with kingdom Monera. As the name suggests, Moneras are nothing but prokaryotic organisms. They are unicellular, only single celled, but prokaryotic. The moment you hear the word prokaryotic, the prokaryotic has a definite meaning. So prokaryotes are the organisms who do not have well developed nucleus, who do not have a well developed nucleus. Here nucleus is absent, but DNA or genetic material is just freely floating on the surface of the cytoplasm. Genetic material is just freely floating on the surface of the cytoplasm. This condition is called as a nucleoid, nucleoid. Nucleus is absent but nucleoid is present. This is the characteristic feature of all the prokaryotes. Eukaryotes, coming to the eukaryotes, eukaryotes have well developed nucleus. What do you mean by well developed nucleus? Well developed nucleus means they will be having nuclear membrane, they will be having nucleolus, they will be having a genetic material within which a nucleoplasm is there. If all these 3 aspects are present within the nucleus, then it is called as well developed nucleus. One is nuclear membrane, nucleolus, nucleoplasm. If all these 3 things are present with, then it is considered as well developed nucleus. But coming to the Monera, here they are completely prokaryotes, unicellular prokaryotes, and they are having a nucleoid. DNA is freely floating on the surface of the cytoplasm. They live in extreme habitats. They can be seen in very extreme habitat like very hot springs, in deserts, in snowfalls, deep inside the oceans. In all these regions we can find these Moneras, or they are also called as bacteria. Their mode of nutrition is always, they may be autotrophs or they may be heterotrophs, or sometimes they may be parasites also. Autotrophs is the organisms which prepare their own food material either by undergoing photosynthesis or by undergoing chemosynthesis. Heterotrophs is depending upon other organisms. Parasite is living inside the body of other organisms. So all these are the different modes of nutrition of Monerans. Their cell wall, very important point, their cell wall is made up of peptidoglycan called as murin. Their cell wall is made up of a peptidoglycan called as murin, along with different polysaccharides may be present, but the important thing is murin. And they have the locomotory structures called as a flagella. They have locomotory structures called as, you might see here, this is flagella here, this is flagella. So they have the locomotory structures called flagella, and their reproductive methods are either they can undergo asexual reproduction or they undergo sexual reproduction. Asexual reproduction, what do you mean by asexual reproduction? Asexual reproduction is a type of reproduction where a new organism is produced from the single parent. A new organism is produced from the single parent. Best example for this is fission. See here, the bacteria is here, it is having nucleoid. When the time of reproduction comes, what happens is part of the DNA starts splitting. Some amount of the DNA goes up some amount of the DNA comes down, and then the two cells get divided, and hence two new cells are formed. It is simple fission. Here no two organisms are involved, only single organism giving rise to another organism. In all the asexual reproductive methods, there is only one type of cell division, that one is called as mitosis. There is called as mitosis. In all asexual reproduction, mitosis cell division takes place. Okay, so these are some of the basic characters of kingdom Monera. See, here is complete entire classification which we are going to study. This is completely broad classification, in-depth classification. This type of classification is not at asked in the examination, but for clear understanding I have given this classification. See here, first kingdom Monera, it is divided into three divisions: Scisiophyta, Cyanobacteria, Protophyta. Scisiophyta, under that Scisiophyte there is a Scisiomycetes class. Under that bacteria are present. Bacteria are placed in Scisiomycetes. This bacteria, it is again divided into Archaebacteria and Eukaryotes. Okay. Next, Cyanobacteria, it is having blue-green algae. Protophyta, they are having Mycoplasma. This is in-depth classification of Monera, but usually it is not at asked, but according to the syllabus, kingdom Monera is divided into Archaebacteria and Eukaryotes. Okay, this is exact classification, but still this is the thing which I quoted now is in-depth classification. Okay. Next, coming to Archaebacteria. Archaebacteria is called as living fossils. Archaebacteria is called living fossils. Why? Because when first earth formed, the first form of life appeared on earth was Archaebacteria. Okay, first form of the life which appeared on the earth were Archaebacteria. Hence they are called as living fossils. Even today they are present and they are having many message within their genetic material. If we analyze the genetic material of this bacteria or Archaebacteria, we will come to know all the aspects of evolution. Okay, hence they are called as the living fossils. They live in extreme habitats. This is the very important point. They live in hot springs, they live in polar regions, they live deep inside the earth. Okay, in extreme habitats they can survive. Their cell wall, see here, very important thing is the cell wall is not having murin, but instead of murin and not well developed murin, that is called as pseudo-murin is present. Pseudo-murin along with many polysaccharides. Okay, Archaebacteria, see here, this classification, bacteria, it is divided into Archaebacteria, Eukaryotes. Archaebacteria are called as primitive bacteria and Eukaryotes are called as true bacteria. Now we are discussing about Archaebacteria only. Archaebacteria, it is again divided into three types. First one is Methanogens, next one is Halophiles, third one is Thermosedophiles. Methanogens are the bacteria which are present inside the guts of cattle, where they help in biogas production, hence methane production, especially the formation of methane, hence biogas is released, hence they are called as Methanogens. They live in marshy area also. Okay, the first one is Methanogens. The primary aspect of Methanogens is they live in the marshy area or they live in the gut of cattle, where they undergo reaction and produce a methane. And also in gobar gas production, methane is released, so that Methanogens are used for the release of methane in the gobar gas production. Okay, so Methanogens. Next one is Halophiles. Halophiles are the bacteria which are present in the salty area, high salty conditions, high salt water. In those conditions also they can survive. Next one is Thermosedophiles, which live in hot springs where the temperature nearly 60 to 80 degrees Celsius. In those conditions also they can survive. Hence they are it is told that they are found in extreme habitats. Okay, this is about some of the examples of methanogens are, see here, example Methanobacterium, Methanococcus, Methanospirillum. Methano indicates methanogens. Okay, Halophiles, whenever in the examination the examples are asked, the example need to be identified based upon their names. Methanobacterium means they belong to bacteria, under the means bacteria belongs to Monera, under Monera they belongs to Archaebacteria. Next one is Halophiles. Some of the examples are Halococcus, Halobacterium. Halo means salty area. Okay, Thermosedophiles, see here, Thermoplasm, Thermoproteus, Thermoplasm and Thermoproteus. Okay, next is Eukaryotes. Eukaryotes are also called as true bacteria. Eukaryotes are called as what? True bacteria. All the bacteric features are present in these group of bacteria, hence they are called as true bacteria. Why? Because they are having murin in their cell wall. They are having what? Murin in their cell wall. They are of different shapes. See here, they are of different shapes. See here, they are of different shapes. Okay, next one is the bacterial cell wall is again having a certain gelatinous coating called as a slime layer. If this slime layer is made up of protein, then it becomes capsule, hard capsule. If it is made up of protein, it becomes an hard capsule. If it is not made up of protein, it is just polysaccharide, then it becomes a slime layer. It gives protection to the bacteria. Okay, as I told, that cell wall is made up of murin, a peptidoglycan. Murin is nothing but a peptidoglycan. Next one is nutrition. The mode of nutrition is heterotrophic. There are some example for this is parasitic. Example, cholera, the disease which is called as choleral. Next one is saprophytes. Saprophytes are the organisms which grow on dead and decaying organism. Example for that is Azotobacter. Next one is symbiotic bacteria. Means symbiotic bacteria are the bacteria which live in close association. Example for this is Rhizobacterium I have given here. See, Rhizobacterium is a symbiotic bacteria which is present in the root nodules. See here, round red color round like structures. These are called as root nodules within which rhizobium are present. See here, these rhizobium are present within the root cells of plant, but they are not giving any harm to the plant. They are seeking shelter, they are taking nutrition from the plant. In turn, they are providing atmospheric nitrogen to they are fixing atmospheric nitrogen to the plant. Hence, it is called as a symbiotic association. Here, plant is also helping the bacteria. In turn, bacteria is also helping the plant. There is no any harm between the two organisms. Hence, it is called as symbiotic association. Okay. Next one, mode of nutrition, autotrophs. There are some autotrophic eubacteria also. Under that, first is photosynthetic. Cyanobacteria. Cyanobacteria are also called as blue-green algae. These are bacteria which are having a chlorophyll pigment. Bacteria which are having a chlorophyll pigment which can undergo photosynthesis. This is very important. Always there will be a confusion between cyanobacteria and rhizobium, symbiotic bacteria. Rhizobium is not having a chlorophyll pigment, but they can help in fixing of nitrogen, whereas blue-green algae, they are having chlorophyll pigment and hence they can undergo photosynthesis. In turn, there are some blue-green algae which can also undergo nitrogen fixation also. Okay, keep this in mind. They are all cyanobacteria. They are also called as blue-green algae. Example for this is spirulina. Nowadays in the market, tablets are coming into the market by the name spirulina tablets. Spirulina is rich in protein. Okay, hence this vegetarian people are having the capsules of spirulina which are high in protein. Next one is some of the examples of cyanobacteria are Nostoc, Anabina. You should always keep in mind, these examples are always asked. Examples of cyanobacteria, Nostoc, Anabina. The question will be there, which among the following are the examples of cyanobacteria? They will be given Nostoc, they will be giving rhizobium, they will be giving azotobacter, and they will be giving another bacteria. You need to pick the correct answer. Nostoc or Anabina. Okay. Then next one is again for movement they have a flagella. For movement they have flagella. Again the reproduction takes place by asexual method. Asexual method takes place by fusion. Fission, as I already told, fission. One cell splitting into two cells is called as fission. Next one is sexual reproduction. Very important point. Here sexual reproduction takes place by three methods in bacteria. That is by transduction method, another one is transformation method, another one is conjugation method. See here I have posted a very nice photo which gives a clear understanding. What happens in transformation is some of the bacterial genome integrates, come and contact with the bacterial genome of another bacteria. It integrates with the another bacterial genome, and hence a new cell with both integrated genome is present. Genome is present within the bacteria. Such type of reproduction in bacteria is called as a transformation. Here the DNA of one bacteria is entering into the cytoplasm of another bacteria by one or the other means. Let's not go into the detail how it enters, but just keep in mind this DNA of one bacteria enters into the cytoplasm of another bacteria, then it comes in contact with the DNA of that bacteria and it integrates with it, and the new organism with the new features are formed. Hence it is called as transformation. Next one is transduction. Transduction is a process where there are certain viruses called as bacteriophages. Bacteriophages are the virus which infect a bacteria. When bacteriophages infect a bacteria, their genome comes in contact with the DNA. This DNA gets transferred to the another virus coming out of this. They take the DNA of this bacteria also. When they go and infect another fresh bacteria, there again new DNA is entering into the cell, so that the cell with a different DNA is getting formed. Okay, this is about transformation, where the DNA is transferring with the help of viruses. Here DNA is directly entering. Next important point is conjugation. So there are R strain, S strain bacteria, or simply positive and negative bacteria. So some amount of the DNA enters into the another cell. By coming close contact, there will be formation of conjugation tube. Okay, and hence as part of the DNA enters into the cytoplasm of another cell. So this type of reproduction is called as conjugation. These are the three important methods of sexual reproduction which is taking place in bacteria. The first one is transformation, next one is transduction, and next one is conjugation. Okay, so here, see here the very nice representation of fission. See here, this is bacterial cell. The DNA got separate, then here the cytoplasm got separate, two new cells are formed. Example of fission, asexual reproduction. Okay, then next one is next group of bacteria called as Mycoplasma. These are the next group of bacteria where the very interesting point is they don't have cell wall. They don't have cell wall. Their plasma membrane is directly exposed. They are not having any proper shape. They are of a different shape. Hence they are called as pleomorphic. Okay, here the main important point need to be considered is plasma are the very smallest organisms. Smallest known organisms are first of all bacteria are very small. Within the bacteria there are certain group of bacteria called as Mycoplasma. These are still more small. Okay, they are very smallest organisms. They are not having any cell wall. Their plasma membrane is directly exposed and they are of a very different shape. Okay, they can acquire different shape throughout their life cycle. Hence they are called as pleomorphic. Okay, this is about Mycoplasma. And the last important point is economic importance of bacteria. How bacteria are useful for humans for the production of or for generating the income. First point is scavenging. Scavenging is nothing but whenever any dead material falls on the ground, immediately it has to be decomposed, right? Or else it will be a very, if no decomposition takes place, then all the dead bodies remain as such. It is not good, and also the minerals, the soil will not get minerals. Hence, scavenging, bacteria will act as scavengers Okay, they decompose the dead and decaying organisms. Next one is they help in fermentation process in the production of alcohols. These bacteria are used. Without methanogens there may be no without bacteria there will be no fermentation process, so there will be no whole alcohol industry, beverage industry is dependent on fermentation. Okay, then fermentation is dependent on bacteria. Without bacteria no fermentation. Even bacteria helps in the production of antibiotics. Okay, so one thing we need to consider here is whenever a dead organism falls on the ground, immediately it will be attacked by various group of microorganisms. Bacteria may attack, virus may attack, fungi may attack, isn't it? Fungi may attack or any other group of organisms may attack. What happens is when this organism attack, this viruses attack, this bacteria and other group of organisms, only limited food is available here, limited dead body is there. So what these organisms do is, while undergoing decomposition process, they release certain chemicals so that they mark clear boundaries among themselves. They tell to this bacteria or this group of organisms that this is my boundary, please don't enter into my area. So even this also releases some chemicals and tells to other bacteria that or other group of organisms that this is my boundary, please don't enter into my boundary. So that they are producing the chemicals. These chemicals are not allowing other organisms to enter into it. These chemicals are called as antibiotics. Anti is life, anti is opposite, biotic is life. They are acting against the life. These antibiotics are extracted. We human beings extract the bacteria and we get treated for any of the diseases. Okay, so bacteria produce antibiotics. Next one is, as I told, many rhizobium bacteria. It fixes what atmospheric nitrogen living within the roots of many plants. Next one is mineral extraction. How mineral extraction are this organisms bacteria help in the mineral extraction. Say for example, dead organism is present, it is having organic and inorganic materials. Organic materials somehow it undergo oxidation, whereas inorganic materials, iron, nickel, cobalt, magnesium and other minerals, they are getting, after decomposition they are getting released into the soil. If there is no decomposition, minerals will not come out, so soil will not get minerals. Hence mineral extraction or mineralization process is helped by bacteria. Next one is in sewage treatment. So all the daily waste which is generating from the home, it is going out through drainages. Those drainages are connected to larger tanks. Those larger tanks haul the big waste materials which can be hand picked are picked out, and it is treated with bacteria to undergo decomposition process, which reduces all the organic waste. Hence for sewage treatment also this bacteria are used. So these are some of the importance of Monera. So one short revision as we have studied. There is what? First is R.H. Whittaker 5 kingdom classification, isn't it? He gave kingdom Monera, Protista, fungi, plantae, animalia, isn't it? Under kingdom Monera, it is again divided into Archaebacteria, Eukaryotes, Archaebacteria, Methanogens, Halophiles, Thermosedophiles. Okay, under Eukaryotes, photosynthetic bacteria. Next one is parasitic bacteria. Next one is
So autotrophic bacteria, heterotrophic bacteria, and this is based upon their shape. There are different types of again eubacteria based upon their size. If they are round, they are cocci. If they are rod shaped, they are called bacillus. If they are spiral, they are called spirulum. If they are comma shaped, they are called as vibrio. Okay, this is about kingdom Monera.